yourself a
All right, I'll go ahead and get started, and then if anyone else comes, they can walk in and everything. So thank you guys for coming. One, taking time out of your day. I was really just prepared. Like, worst case scenario, I was like, Augie, Taylor, Sarah. Boom, that's what I know I'm presenting to and everything. So it's cool to just have a few more people in here. So this backstory of this was my final project and presentation for my class this spring in biochemistry. And as we've all known, I've had conversations with a few people here this semester. These are not the most easy of applicable sciences out there. You know, we read this stuff in textbooks and we're like, it all sounds really cool in the moment, but it's like when we're sitting down and we're programming as trainers, it's kind of like all that stuff kind of sits in the back of your mind, but like kind of doesn't at the same time, you know? Like you don't really need to know how much NADH and FADH is created in the Krebs cycle to sit there and tell someone how to do a Bulgarian squat correctly, you know? And one thing that really got me excited for this was when I learned the term of what Lauren likes to say is you're not a full master of information until you can explain it to like a 7-year-old basically. And so this is pretty intensive information in there. There's going to be a lot of different letters that are thrown together and you're like, that doesn't actually spell a word. And you don't really have to remember every hormone per se, but I like to look at the trends. You know, I like to look at the things that kind of start to put together, and I wanted to try to explain this in 3 different avenues. One is the academic avenue, because this was for a grade, so that's where the depth of the knowledge will be. 2 is how can we apply it to ourselves, you know? Like how can, as a trainer, if I understand this information fully, how does that make me a better trainer? And 3 is if I have an athlete that doesn't know squat about science or the body or anything like that, but they really are understanding, or maybe they come to you and we're doing sprints for conditioning and they're like, coach, I thought I had to run a mile and a half to get conditioning, you know, and we're only sprinting 3 times for the whole hour and you call that conditioning. Like, how does that work? So by understanding the basis of how the body works and all this, this will kind of give us a key and a guide to why we do what we do. And it's really helped me actually watching Augie, how he goes through the conditioning of our elite group in the morning and everything like that, until I kind of understand, oh, this is how this works. Like this system's starting to kick in more than this system, and these are things that are starting to happen, and it all really starts to make sense when you think about how the body works 2 to 3 weeks of consistent training and everything. So the way that I like to describe biochemistry is I like to use an analogy of, say if I move to a new city and I'm new in town and I'm like just decide, hey, I want to be the best party thrower out there. Well, I'll throw that first party. I'll do things that I think are good, that'll catch a vibe, that will do all these things and make everyone happy. But that first party is probably going to be the worst one that I ever throw. There's a lot of things that could go wrong. I could pick the wrong music. I could pick the wrong food. I could pick the wrong drinks. I invite the wrong collection of people that just don't match well together. Well, it's like, all right, now next Friday I'm gonna throw another party and it's gonna be better. And with every single weekend that I decide to throw a party, I get better as a host because I've learned to adapt. I'm like, okay, I'll do this differently. I'll do this differently. I'll do this differently. And that's how I like to think how the body actually works through exercise. The very first time we go into an exercise and we introduce an environment, we're like, this sucks. Like, I don't like this at all. And then now with every single time we decide to introduce that stimulus again, the body makes little tiny changes that will sit there and be, okay, let's get better, let's get better, let's get better. And these are all the little hormones that kind of uptick, downtick, how things start to change to better that environment so that we become better athletes. So, sprinting, any exercise in general, but really what happens is all the arrows kind of point and tell a story. So for us to sprint, our brain sends a signal to the muscles, and this will tell us to contract. This will tell us how hard to contract. This will tell us what exactly our body needs to do, and the muscles will take care of the movement. And while this side is happening, our muscles need things to work. They need nutrients, they need oxygen, they need all kinds of things to sit there and keep going. And so that's where the heart and the circulatory system comes in. Through our transportation of blood through the arteries, it sends all this stuff to our muscles that allow us to continue this. And then also at the same time, it sends all the negative stuff away. And it allows it to go through its other systems and everything and be like, okay, this is cool. Like, you know, exercise is hard work. We're going to have waste and we'll be able to do different things. So for us to be able to understand chemistry, biochemistry, we have to understand a little bit of the physiology. So right here, I just have a very simple paraphrase of the cross-bridge cycle. And as we all know, this is a cycle that must occur for our muscle to contract. So the reason why I wanted to bring this up is because based on how much this contracts comes from calcium release. Calcium release is the first hormone that's really important when it comes to biochemistry of sprinting. So to break this down, this is that neuromuscular, from the brain to the muscle signal I was talking about. Action potential occurs right here at the neuromuscular junction. At that action potential, now it goes down these little pathways. These little pathways is what depolarization is. Depolarization is simply from potassium and sodium. These are our electrolytes. I'm sure we've all heard of electrolytes before. These two electrolytes are the main things that occur when it comes to muscle contraction. The way I kind of like to think about it is like, say these are like two like rival high schools, and now it's like, hey, we got to try to become one as a city or something. And you sit there and you cross these walls, you cross these boundaries. That's what potassium and sodium is doing. Well, what happens is, let's say we throw everything in flux too much, everything's tight, there's a lot of tension. It's like, all right, we got to go back for relaxation. So that's where repolarization happens. And so everyone will go home to their natural states. So that's the very first thing that will start to happen with depolarization. So as its action potential goes down to the T-tubules, it'll hit this guy right here, the sarcoplasmic reticulum, and this is the storage place for all calcium. So like I said, calcium is extremely important for contraction to happen. So calcium gets released, and then this is where the cross-bridge cycle actually starts to occur. Calcium binds to troponin. Troponin makes these little Twizzlers go away, opens up these holes for actin, and then now the myosin head will be able to go and connect to the actin. And I'm sure this is a lot of review for you guys, but I just want to make sure that we get this down before we go into a lot of the deeper stuff. So ATP is used in 3 different phases. As we all know, ATP is adenosine triphosphate, which is just energy. And the way I like to think of it is like, imagine like 3 balls that are all connected together magnetically. ATP is 3 balls. Well, now every single time we use ATP, ATP is used at 3 different places. It's used for relaxation. It's used to put calcium back into the sarcoplasmic reticulum. It's easy to come out, but it's hard to put everything back in. And ATP is used for potassium and sodium. So in just this one specific sequence, ATP is used in all 3 of these things. So now we broaden our horizons a little bit. So the different colors of the arrows will now start to tell the levels of the story. So if I come over here, when we exercise at higher intensities, we need to ventilate more. Do we need to bring oxygen in? And I know this arrow right here shows that oxygen is down, which is actually not the case. We're going to take in more oxygen when we sprint compared to when we're at rest, but it's the significance of this oxygen. Oxygen does not cover all the bases when we do these sprints at the same way that it does in resting. So even though oxygen levels go up, the importance of it goes down. The lungs start to come in, and this is where air displacement starts to happen. We talked about that waste that occurred with the muscular system. So oxygen comes into the muscle system, and then it starts to put out carbon dioxide. Carbon dioxide enters through the veins, and then the vein goes back into the lungs, and that's where we breathe it back out. That's our first mode of disposal of waste in the body. So now we go back over here. This is the cross-bridge cycle, this thing we just talked about. When we're sprinting, we're having more contraction. So this level goes up. If this level goes up, calcium's going to go up because calcium is the thing that causes contraction. That's important. We'll get back to that later. So now we get over to, we talked about how ATP is used. We got to talk about how it's formed. So we have the phosphocreatine system right here, and then we have the Krebs cycle right there. One thing also, Lauren kind of talked about this last Saturday, is bioenergetics is not as simple as we sit in one zone and that's the only energy system we use. Actually, when we exercise, all three energy systems go up. Our, he calls it alactic, lactic, and just like oxidative. I'm going to stick with phosphocreatine, glycolytic, and oxidative. So this is our phosphocreatine system. We'll talk about this in a second. This is our glycolytic system, and then we have our oxidative system as well. But this goes down in significance. The same thing with oxygen, because its role is not important when it comes to sprinting at 90 to 100%. And that's what this whole topic is about. And then we'll see right here, creatine kinase, that goes up as well. So we get into the phosphocreatine cycle. So we talked about the three phosphate groups that are energy. We have ATP, ADP, AMP. So they're just three balls that are connected magnetically. And what happens is when we use ATP, it's broken down by one ball at a time. So ATP and water are connected through ATPase, which is an enzyme that breaks down ATP, and it gives us ADP and inorganic phosphate. So our three-ball molecule is now split off, and now it's just two balls. These two balls right here do not hold the same weight, and they cannot do the same amount of things as the three-ball molecule can do. So in times of extreme ATP replenishment, what we have to do is we have to use creatine kinase, which, as we all know, is a supplement that's widely debated and everything. That's probably the first supplement that young athletes will kind of come asking us for questions like, Coach, do I need to take creatine? Probably as a young athlete, probably not. But creatine also is not evil. It's not bad. There's side effects to it, but ultimately, in times of high phosphocreatine cycle usage, it is a supplement that can help us. So that is something that we can take as practitioners, is if we get these elite level athletes and they're looking for that edge, and maybe we've already addressed their sleep, maybe we've already addressed their nutrition, maybe we've already addressed the amount of proper work that we're doing, and they're still looking for something, that's, in my opinion, where we should start to get into supplementation. Supplementation is more for fixing the curves or everything around the edges. It doesn't fix the core of the problem. So ATP is broken down through hydrolysis. This is the first biochemical term. Hydrolysis is basically just taking hydrogen and using it to break apart a bond. So as we see right here, water has a hydrogen molecule in it, breaks it down, ADP, inorganic phosphate. Inorganic phosphate is waste. Waste is something that we got to get rid of. So creatine kinase allows for the buildup of ATP during exercise. So this is really quick. It's the most efficient mode that we have, but it's not injury, or it's not fatigue resistant. We get tired really fast when we use this, so it's not something that can occur for long periods of time. One thing also I want to talk about is this is all for in times of exercise. Whereas when we stop and we rest, so we sprint someone for 10 seconds, I think a lot of times textbooks teach us what energy systems happen in times of exercise, but we never think about what results from exercise, if that makes any sense. So when we sprint for 10 seconds, it's not that 10 seconds of work is the only thing that's happening for adaptation to occur. It's, all right, 45 seconds down, 90 seconds down. Whatever your work to rest ratio is, your oxidative phosphorylation and your glycolytic system, they're both ramped up. Like we said, during exercise, all three systems are working because we want to meet ATP needs as much as possible. So that's where we start to get into things. We talked about ATP usage earlier. All three of these things are working to find us homeostasis, to find us balance. 70% goes into the actin head contracting and relaxing. That's where the majority of our ATP is used. Whereas with the SR, so calcium cycling, and with our potassium and sodium cycling, getting that balance back at all three of those modalities is what the muscle has to do every single time you go into contraction. So, PCR restoration. This is how our PCR system works. Like we said, it is highly efficient, but it doesn't look like it here because we just crank through all this creatine that essentially happens in the body. Whereas it starts to recover over time. You see from 30 seconds, 60 seconds, 120, and eventually there's research out there that says 4 minutes is needed for us to actually complete or replenish all of our PCR that we just used. Which, as we know in the field, we don't have 4 minutes to sit there and spare between every single set of something, especially if someone's paying us for only an hour, we got to get in and out. So this modality starts to go up, but this all happens during EPOC. Essentially, it's this picture right here. Hands over the hips, you're breathing heavy, breathing hard. During that time, our body is now recovering from all the stress that we just did. And that's really important when it comes to sprinting at high durations. We put everything in the most stressful, metabolically demanding activity that we can possibly do. And now we deplete everything and we're like, hey, get us back. We got to sprint again. So I have a question is like, if, say this is an untrained person, okay, someone who's never sprinted before, and then I get an athlete in here that's been doing sprint interval training for 10 weeks. Does this line go down faster or does it go down slower?
For the known athlete?
For the elite athlete.
Faster.
It's going to go down faster. Does anyone know why?
More efficient at using those systems?
You're more, not the systems. If I'm bad at sprinting, and we think about mechanically, for example. If I have bad mechanics and I don't push, and then we fix everything to become more efficient at moving, what is happening? What am I doing? Intensity is higher. How do I raise intensity?
Speed, more efficient.
Speed and force into the ground. If I'm required to put more force into the ground, that means I'm activating more muscle fibers with every single contraction. So if I'm able to use my body more efficiently at higher rates, that means I'm depleting the system as fast as I possibly can because I'm requiring more demand of the cell. But at the same time too, if I'm more trained, this is going to go up faster. That's going to be much steeper to go back. And that's the very first thing we start talking about is how can sprinting get me more in shape? One is I'm better at movement. Two is now I can recover from movement. And that's probably one thing I think we can tell young athletes right there is like, coach, how does this get me more in shape? I only sprinted for 6 seconds. It's like, we're teaching your body to be as efficient, as powerful as possible. And then this is a little bit more advanced, but it's like when we get as powerful as possible, our body has to recover. And so it recovers faster so that we can do it all over again. So, lack of oxygen. During our sprinting, we surpassed the respiratory exchange ratio, RER. So when I did a lot of testing in the lab, I probably tested about 50 or 60 VO2 maxes. And if you guys don't know, that's where the test where we hook up of a tube to their mouth. We have them sprint using different tests, and it basically tells us how much oxygen someone breathes in versus how much carbon dioxide breathes out. And that is our respiratory exchange ratio. So when I was in the lab and I had someone hooked up to the machines and I had all these numbers in front of me, there was one number I watched as the tester to know it was time to go. It was time to start putting the gloves on. I'm getting ready to clear the test, wipe down the machine. It was when that number got above 1. And so this is something that is very high advanced. If you ever worked with an elite level athlete and they wanted to get tested, that is something you can actually look at is, okay, when did they hit 1 on their RER? Because now I know that maybe they hit 1 at 7 miles an hour on a 1.2 grade slope. It's like, okay, now when I'm programming, and if I want to get that specific with my speed and I want to control as many variables as possible, it's like, I look at the speed, now I know that at 7 miles an hour, they're using this energy system versus this one. They're able to be sustained or they're going to get really tired. And then now that tells something, if you have the modalities to do a VO2 max, the very beginning, you test when they hit that RER. And then at the end, you can also sit there and be like, wow, okay, you were able to maintain speed at 7.5 miles per hour at a sustainable level. So that means during performance, they're running faster with energy efficiency, essentially. And then you can also look at the majority of VO2 max, which is just ventilation, how much oxygen you're breathing in versus how much carbon dioxide you're putting out. So this is something I also want to talk about is in high intensity, short duration exercise, like I said, we don't have a lot of oxygen. We're putting way more out than we're taking in. So this will mean that there's going to be a little lactate production. And spoiler, we'll talk about lactate in a little bit, but lactate is always being produced. There's research out there that shows that lactate is actually produced when we're aerobic. And so that's one of the things I really saw. I was like, okay, wow, that's different than what we've kind of always been told or research has showed us. So now the diagram expands. We've now moved on from the green arrows to the pink arrows. So we talked about how the significance of oxygen is lower. So this opens an entire cycle over here on this right side that shows how to be more efficient with our oxygen. And over here, we just went over the bioenergetics where ATP levels are down because we can't replenish them, because we are just at too high of a demand. So our ADP levels are going to go up and our ANP levels are going to go up. And that's really important for a later cause to show us which type of hormones start to get activated because of the ratio. And that's one thing I've also learned about the body is everything's in ratio. It's not one thing beats over another. It's, okay, when we're at 55% pyruvate versus 45% lactate that's available in the cell's going to be in one type of environment. But now, if that number gets switched, all other types of things start to work. So, hypoxia inducible factor 1A, HIF1. This is where we start to get into all the letters and everything like that. Don't worry about the letters, just know that they play an important role and they start a trend in the body. So HIF1A is available in only hypoxic environments. So this is one cool thing, is this is how the body shifts. Only in times of sprinting or high intensity duration, and we don't have a lot of oxygen that's available in the cell, HIF1A is available only in this moment, and what it does is it's a ubiquitous trans factor. So basically, this is something that kind of sends a signal, and ubiquitous means breaking down. The way I like to think of it, it's like almost like a dishwasher. If I sit there and I take a plate and I put it in that dishwasher, the dishwasher is going to cut off all the dirty food, cut off all that waste that I don't need. That way, it winds up coming back out on top, nice and clean, nice and efficient. The plate in that analogy is a protein. So, how proteins work is, if, say, this gym was empty, and we were like, all right, we all got a session in an hour and a half, and there was a big product, a big package that entered those doors, and every single one of us had an athlete, and we had things that we needed to get done in that next hour. What we do is we take all parts of that package and we build it for our specific purposes. That's what protein synthesis is. We digest protein, it enters the gut, it gets broken down to all these amino acids, these little blocks, and now all different parts of the body is taking these blocks and it's like, hey, I have a need, I have a desire, so I'm going to create my own proteins to fill that desire. That's how proteins work in the body. So that's why some of them have more Legos than another of a specific type. So that's why we get to the difference of maybe like whey protein versus plant protein, because these types of proteins offer different things that the body needs during specific exercise. When we work out aerobically, that's way different than when we work out in a resistant exercise. So our body has different needs, so we need different proteins because they give us different blocks. So ubiquitous HIF1A starts the degradation, so the breaking down of all this protein that's damaged. We clean off all the damaged part, so now protein synthesis can start to kick in. Right here, HIF1A regulates anaerobic metabolism, and it also has a half-life of less than 5 minutes. A half-life of a protein basically is how much time it takes to die down. Lauren talked about half-life a little bit in his talk about just power, strength, speed. You know, there's only a certain amount of time a body can hold on to a stimulus before it just can't keep on to it anymore. So this is really cool because now if I think about it, in times of sprinting, HIF1A is super high. And then the second I stop sprinting and I reintroduce oxygen back into the cell, back into the body to a substantial level, it starts to die off. We switch from anaerobic metabolism to aerobic metabolism. This is mitogen activated protein kinase, MAPK. it's just different pathways. I just put that in there just to kind of show that the body's super complex, and there's a lot of things that go on to it. But I wanted to pick out these specific proteins that kind of help show the regulation of everything that goes down. So, HIF1A, sorry, down here you can't see, it allows for more new cardiovascular tissue to be created for more dependable oxygen transportation. So HIF1A, if we look back onto this thing, it connects to this guy over here. And what that guy is, that's VEGF, vascular endothelial growth factor. Endothelial, if you guys remember from AAP days, it just means tissue. So, so vascular, part of the system that transports blood, endothelial growth factor. It's going to grow this tissue. HIF1A starts the activation of VEGF. VEGF will now go into the artery and it will start to build capillaries. That's what's called angiogenesis. And the way I think of angiogenesis is if I'm on a highway and the highway's really backed up, I will build more exits on that highway to allow things to start to get off. That's what capillaries do when it comes to the circulatory system of the body. It allows all these little mini pathways to enter from the blood, so all the nutrients that come from the blood, they can get into more muscle cells. So by sprinting and introducing our body to no oxygen, our body's like, hey, I need this stuff. I have to have it to sustain life, so it does everything in its power to create a more efficient system for oxygen. As we talked about with oxygen and bioenergetics, that's where our glycolytic system and our oxidative system. So when we go back to rest and we allow that reintroduction of oxygen, our body is now more efficient at using whatever it's getting to grasp back onto life and to replenish ATP, because we need ATP to complete muscle contraction. So that's what this whole side is. It's our body's ability to use whatever oxygen we get more efficiently. So now how do we use that? That's where this blue side comes into play here. Mitochondrial biogenesis. So we've all heard the mitochondria is the powerhouse of the cell. Does anyone know why it's called the powerhouse of the cell? The mitochondria is the holding place for all of our bioenergetics. That's where phosphocreatine system lives. That's where the Krebs cycle lives, our glycolytic system. That's where our oxidative and Krebs cycle are kind of together. So that's where all of that happens. So essentially, in the most simplistic way, is if I can create more mitochondria, I create more sites for ATP replenishment. So that's how all this is going to start to get kicked in. Don't worry about all those words. Basically, by the MYOD proteins, it's just a family of proteins. It'll start, I just put this in there because it just wanted to show the whole number of steps that the body has to go through. So nuclear respiratory factors, we all know respiratory means oxygen, carbon dioxide, ventilation. So by our body's ability to use this stuff, it'll start to activate other things. So it leads to transcriptional currents controlling mitochondrial biogenesis. Biogenesis just basically means the creation of something in the body, essentially. So mitochondria, we're creating mitochondria in the body. NRF, these factors connect to the MTFA or the TFAM over here, kind of similar. And so now it will start the upstream effect of NRF peroxisome proliferator activator receptor, PPAR. Not as important for our discussion today, but it also activates the gamma coactivator PGC1. PGC1 is huge, and we're about to get into that. Upstream, that's something that is important just in biochemistry in general. Upstream versus downstream, it's exactly what it sounds like. Think about like swimming in a river. If I swim downstream, a lot less energy. I get there more efficiently. If I go upstream of the river, I have to spend more energy. So it costs ATP for upstream activation of certain things that happen. So, peroxisome proliferator activator receptor coactivator 1A, PGC1A. This guy is just the mastermind of all this. This is the biggest thing. A lot of things activate this guy, and this guy does a lot. It's tuned to reflect the cellular energy demands with conditions of increased demands. So we already talked about sprinting, the most metabolically demanding thing we can probably do in the body when it comes to performance. So if we do the most demanding thing, this activation gets spiked like crazy. It's like, hey, I gotta meet this. If I don't meet this, my body's gonna fail me. So this is just a lot of the things that it starts to activate and a lot of things that come back and activate it. Just know that this is like the parent of all these little children. PPAR, PPARA, ERA, FOXO1, HNF, NRF. That is just all of those things that help this. It regulates all gluconeogenesis, fatty acid synthesis, oxidation, and glycolysis. So it regulates our ability to take things that are not glucose, not carbohydrates in the body, and create sugar from our liver, from our muscle. That's the first thing it does. It helps from fatty acid synthesis. So it takes the fatty acids from our adipose tissue, and it helps build glucose, glycogen, muscle glycogen, for our muscles to use that for contraction. It helps with oxidation, so breaking things down with the use of oxygen, and glycolysis. We all know glycolysis, carbohydrate breakdown, muscle glycogen breakdown. So we see all these signs right here. PGC1 helps oxidation, helps lipolysis, brings down inflammation. All good things. Everything right there is a good thing when it comes to performance. So how do we turn it on? The demand of calcium. Remember calcium from the cross-bridge cycle? We use it a lot for a lot of muscular contraction. So this right here from exercise, cyclic AMP is a secondary messenger. Calcium right here. It will uptick and it'll start this entire process that will go to PGC1A. As we can see, the cold will activate PGC1A, exercise itself, P38 AMPK. Simply, that role is in glucose metabolism and energy expenditure. We break down carbs when we exercise. That's metabolism and energy expenditure. We talked about ATP breakdown. We have to build it back up. So this guy has a major role in all of that as well. So VEGF, we talked about that a little bit earlier with the building up of capillaries. So when we increase this, we increase calcium, we increase this. These are all things that we can do to increase PGC1A alpha. Right here, we have an arrow right here. If you can't see it, it's up of AMP, ATP's down. That's the ratio I was talking about. If we're in times of extreme fatigue, that means we don't have a lot of ATP in the body because we're too busy trying to build it back up. Sometimes if we put increased demand of ATP, that 3 ball gets broken down to 2 ball, and if we're still demanding, we're still telling our cell, hey, we need this, it gets broken down to AMP. So now we have 1 ball when it's supposed to be a 3 ball system. So all of that, NAD, NADH, if you remember from just glycolysis breakdown and everything like that, just know that those are important in there. Those signals go up and down, and all of this reverts back to PGC1A alpha. Calcium breaks down to 2 other things. These are transcriptional factors, so they're basically just sending a signal. It's like, hey, we don't like this, boom, make this change. And it's an auto-regulation loop of everything that's happening. Once PGC1A alpha gets going, everything else gets going. There's upstream effects, there's downstream effects. And it's basically, like we said, this activation shows us what the cell state is. And if the cell's in a bad state, it's going to do everything possible to keep activating this because this will be our lifesaver when it comes to extreme energy expenditure. This is just another slide basically showing everything that happens. Insulin, cytokines, exercise. We get these 2 guys. These guys go through all their little routes, bam, 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 all the way back to PGC1 alpha. It just goes to show how important this molecule is and just show, represent the body and everything. AMP activated protein kinase, AMPK, just like we talked about already, the AMP to ATP ratio. So the more we put our body on fatigue, the more different types of synthesis or rates of just activation of certain hormones and molecules, they're going to start going because our body's like, we don't like this, we got to get back to it. And these are all things that are happening in the resting state when I stop sprinting. Yeah, nothing too important with that. So, citrate synthase. Citrate synthase is just it's a biochemical marker of Krebs cycle activity. So remember, Krebs cycle activity is just replenishment or breakdown of carbohydrates for replenishment of ATP. It's our body's way of saying, hey, we got to use this stuff. We got to get more of it. So if y'all remember the Krebs cycle, I don't have a diagram in here, but it's like one big circle, and it's like a lot of different things that are happening and all different types of products. I don't know about y'all, even through my graduate program, we still didn't go into all those hormones. They're just like, just know that this does this. And citrate synthase actually sits on top of that circle. And a lot of researchers have used this specific molecule to see what is the state of that. How fast is it working? So, Dr. Bishop, he's someone I really enjoyed reading a lot when I had to come to all of this research and stuff. And he had a crosstalk. He was with two other professors. And what they did is they literally just reviewed all of these exercise modalities, and they wanted to see what's more important: exercise intensity, exercise volume. In the grand scheme of things, they found that volume is the most important thing. This line down here is volume. You'll see we have much higher rates than most of our low volume things. So in terms of adaptation, to really get our athletes the most in shape, volume is ultimately the number one thing that will win overall. But as practitioners, we know it depends on your sport. It depends on what exactly their role is on the team, all this stuff. And my argument is that intensity can get us in shape. And the only reason why I bring this up is we look at 4 modalities right here. These circles, if you can see them, is sprint interval training. Our triangles is HIIT interval training. The X is moderate intensity circuit training. So think of like your CrossFit, I guess. So like your moderate intensity plus HIIT together. And then the little diamonds is just moderate interval training. So think of it like fake HIIT, like people think they're doing HIIT, but they really aren't that intense or anything. So if you look at volume, we actually don't see a circle all the way till back here. So all of these studies to the right of that imaginary line are other modalities that have more volume. But the one I want to show is, look how many circles are above this threshold, which is proper citrate synthase activity for their research, with how little volume. This shows me right here that from the demands that we put on the body from sprinting, even with extreme low volume, we are getting the demands just as much as this HIIT training, this moderate interval training. So in times of efficiency, especially when it comes to we only got an hour, bang for buck. I can get a lot of bang for buck if I just sprint my athletes versus telling them to go and do something aerobic for 45 to 50 minutes. That's great for athletes. That might even be great for gen pops. If we can get a gen pop in here that can actually sprint, their body can hold up with it. Because a lot of times, if you get people that only want to exercise for fitness, they want to do everything efficiently, as quickly as possible. So to me, that shows that sprinting is a proper modality to get adaptation. So that's really everything that goes on in the body for adaptation to get in shape. We talked about how our body uses our phosphagen system better. We're better athletes. We use energy more efficiently. We recover more efficiently because we put our body in such demand that we have to get back. It's like, I don't like this. I'm going to do everything I can. Well, how do we get better demand? Or how do we get better recovery? It's better utilization of oxygen. We increase our little traffic exits to allow red blood cells to exit the artery to go into the muscles, to get into the sac where gas exchange happens at the muscle fiber level. 3, mitochondrial biogenesis. I like to think of mitochondrial biogenesis as, imagine if a city opens more power plants for the neighborhoods. You don't have one power plant whose one job is to control the entire city's energy production. Now we got 4 or 5 of those things, and they can all start to spread this energy more efficiently. I wanted to end just with a little bit of discussion on lactate, because lactate is probably the most misunderstood thing in our entire field. And there's just a lot of research that's been out there recently that has shown that lactate is, while too much of it is not a bad thing, there is correlation of too much lactate accumulation just corresponding with just fatigue in general, but it's not the cause, it's not the root. Lactate actually winds up being the most, maybe not efficient, but the most prioritized energy substrate. We actually, in high demand exercise, we use lactate more than we actually use pyruvate, because we have so much of it in the body. And I'll go into a little bit about it and how our body actually uses it more differently than we originally thought. So lactate is a product of glycolysis. We all know this. Breaks down, lack of oxygen, pyruvate gets turned into lactate, bam. We've been told that our whole lives, or educational lives. So one thing that was really cool that I first noticed, or first read about, was it's called the lactate shuttle theory. It's by Dr. George Brooks. And I just read an entire 25-page paper on this stuff, and it's pretty cool. But I'll just take out some highlights that I thought were important for our purposes. So we all know that we have type 2 muscle fibers and type 1 muscle fibers. Fast twitch, slow twitch. We all know that fast twitch is very fatigable. Slow twitch is not fatigable because of our ability to use, to create ATP, for our ability to use oxygen, for our ability to produce force. We all talked about those things already. So what actually happens is there's intracellular transporters that take the lactate from our fast twitch muscle fibers, and it will actually transport it over to our slow twitch, to where our slow twitch are not really being used to the high extent during exercise, but now it's taking this lactate to use it as a substance for glycolysis, as we know will create ATP and everything. So now we have this big talk of like lactate appearance. So we all know, if like, I'm sure we all know, if you prick your finger and you measure someone's blood, it'll show you the lactate levels. Obla, onset, blood lactate appearance. I'm pretty sure that's it. I could be wrong on that.
Is it accumulation?
Accumulation. So lactate appearance is actually that number we see right there, but there's actually lactate disposal. So as I mentioned before, lactate actually can be produced in an aerobic setting. It's been proven, it's been shown, because of mitochondria respiration rates versus oxidation rates. Sorry, I forgot that one. But basically is by how much availability of oxygen, things like that we have that occurs at the mitochondria, we also have an amount of stuff that has to get produced out too. So because of this ratio, it has actually shown that lactate can be produced in a fully aerobic setting. And that's kind of like the first clue, is like, okay, if it's produced in this, there might be other things that kind of go on with it. And lactate disposal is all the stuff that occurs through ventilation, because we actually breathe lactate out when it goes into all this different systems. Through the Cori cycle, this is where gluconeogenesis comes in. If you guys don't remember, lactate is a carbon-based skeleton, so just like pyruvate, the only difference is a hydrogen ion is added onto it. So it can take the same backbone, the same skeleton as sugar, and can actually transport it into like muscle glycogen, or muscle glucose, which gets turned into muscle glycogen in the muscle, from the blood to the muscle. Beta oxidation and glycolysis. Now we've taken lactate either in the slow twitch muscle fibers or through the liver, and now we have put it into beta oxidation or the glycolytic system, depending on intensity and amount our body's having to work and everything. So this is what the lactate shuttle is. We have our fast twitch muscle fiber, our slow twitch muscle fiber. Glycogen gets broken down into lactate, has two pathways. And the new stuff is the fact that one is, there's been research that because biochemistry kind of goes with the technology that's available, there's technology that now shows us all these little tiny hormones and things that are available at the cellular level that we didn't know before. So there's been MCT1 is a big one. That's the transporter of lactate that shows us things can happen from cell to cell, where we originally thought that if any lactate was produced, we had to get rid of it, so it went into the circulatory system. So it actually gets moved over to our slow twitch muscle fibers, goes into all these little mitochondria, and now that can get turned into glucose or glycogen, or it can be used to create ATP essentially. Carbon dioxide is waste. We talked about this before. Now all this blue is our venous system, so lactate and carbon dioxide do go through, and this part is all true, it goes through the lungs, it gets breathed out. Lactate is actually a substrate that we use for our heart, for our brain, for our liver, and for our kidneys. So in times of high intensity exercise, lactate is actually the primary substrate that all of those things want to use over muscle glycogen. Even during times of re-eating, or after we've fed ourselves, because we've also thought that all glucose or muscle glycogen goes straight into like the cell, straight into this. There's actually a kind of like disposal rate, like 20% of things that we take from food actually bypass all of this and go straight into the muscle fiber cell, which we didn't know before. So this is the journey. I'm not going to get too much into it. Just know that this is happening at the cell. We have the outer membrane of the cell. We have the inner membrane of the cell. MLDH, this is lactate dehydrogenase. So we talked about earlier, hydrogen is used to break down bonds, all these little things that happen. So lactate can be hydrogenized, which we didn't know before. So it takes these little hydrogens over here from NAD, adds it over there, takes it away from lactate. MCT1, that's the other major component that has been kind of shown that the lactate shuttle theory works. And this will carry it across the inner membrane. So lactate is now across the inner membrane, been turned into pyruvate, and enters the citric acid cycle, Krebs cycle, whatever you want to call it. So this just shows from glucose to pyruvate. We don't have oxygen. Oh shoot, so we take a hydrogen from pyruvate, we put it over here on lactate. Lactate crosses this outer membrane. That hydrogen now gets taken away. The one we added on there gets taken away, boom, turned back into pyruvate, which is something that we did not know for a long time. These are really small. Like I said, we talked about lactate appearance versus lactate disposal. Lactate doesn't increase at workload. So if I'm more trained, it doesn't mean that I produce more lactate, or I produce less lactate. My disposal rates get higher. I'm able to transfer it over from muscle cell to muscle cell. I'm able to use ventilation more. My kidneys, my brain, all this is able to use it more efficiently. So that's why if I sit there and I run something at a certain intensity one day, I'm untrained. I train for 6 weeks. I go back and I do the exact same workout. And if I were to measure my lactate levels in my blood at the very beginning versus the end, it's not that less is being produced. It's I'm more efficient with how I use it in the body. Lactate is a major fuel source. So we talked about the lactate to pyruvate ratio. Said it's not 100%, either way. There's a ratio of these things that happen, and that ratio winds up changing the system of the cell, the system of the body. During exercise, the brain, the kidneys, and the liver all use it for fuel. Lactate becomes the primary fuel source during high-intensity exercise for some cells. Lactate's also been shown to increase oxygen transportation. We talked about increases angiogenesis, VEGF. We talked about that way back. Lactate actually helps stimulate all that. So now our body's like, okay, let's get more oxygen in here. Lactate also increases brain function. It goes neurogenesis, neuroprotection, neuroregeneration, synaptic plasticity, formation, retention, recall of memory. BDNF, I can't remember the exact word for that, but that's actually a hormone that's really researched a lot in brain function and just like older individual, things like that. That's like the hormones, like, okay, is the brain working? And lactate actually has been shown to increase that. And lactate has also been shown to be really powerful in maintaining cancer in some cells because it's such an efficient fuel source that there's treatment out there for some cancer cells that they block lactate because lactate is super efficient and they want to kill the cancer cells as quickly as possible. This is paraphrasing a lot right here. I'm just kind of putting out there what some research has shown us and how lactate can be used in the body, and it's not the boogeyman or the thing that we've always thought is evil. And then the astrocyte neuron lactate shuttle. This was one of the very first things that showed that a neuron can take lactate from the bloodstream and use it as fuel instead of pyruvate or glucose. So in summary, this entire talk, I'll break it down to 4 points. More trained sprinters and athletes are more efficient with their PCR system. We talked about they got higher levels of creatine kinase, so they can replenish ATP more. More muscle activation. They're stronger, they produce more force, they produce force faster. They have higher rates of the cross-bridge cycle. So all 3 of those things go into more efficiency of our phosphocreatine system. Introducing the body to a state of hypoxia increases our ability for O2 transport. We don't like not having oxygen, so the body will do everything it possible to increase these pathways to get us there. Talk about HIF1A, VEGF, increase mitochondrial biogenesis, more specific sites for ATP replenishment, more energy creation, lactate handling. Lactate goes back into mitochondria, all this stuff. We talked about the different transportation from the cell to the cell, or outer membrane into the inner membrane. And our ability just to use it more efficiently through ventilation, through circulation, through gluconeogenesis, and through the lactate shuttle. This is my final diagram. It's not 100% or anything like that, but I just thought it was really cool. We talked about how all these different things, it leads to a puzzle. Whenever we sprint, we put our body in a state of depletion, in need of restoration, and all of these things kick in because it doesn't like that state. And that is what happens when we introduce our body to the most metabolically demanding thing, and it'll fight back, even in short interventions. I just want to thank Lauren set me up with Carl Valley. He's an author at Simply Faster. Really smart guy. I actually only talked to him for like 15 to 20 minutes, but he gave me so many gold nuggets to look into. When I first started this, I honestly knew nothing about biochemistry, and I was like, shoot, okay, like I don't even know where to start. It all sounded really confusing to me, but just in that 15 minutes alone, he was able to give me about 5 or 6 just different ideas. He told me about David Bishop, really good stuff to look into. If you guys want to read any of it yourself, it's a lot, I won't lie to you, but David Bishop has some good stuff about lactate handling and high intensity exercise. And George Brooks, he is basically the main forefather of the lactate shuttle theory. He's got 40 or 50 years work of research about lactate in general, but he broke it all down to like a 20 to 25 page paper. It'll take a lot, but it'll really teach you a lot of stuff, and it's a very controversial theory, and a lot of research has been out there to fight back about it. I've read other lit reviews about it where they read the research that tried to debunk it, and they read his research that showed that it was true. And from what I've read, probably about 60 or 70 pages of all that, I believe that it is used, and I believe that it'll start to be more profound in research and school teaching, all this stuff. And that's it. These are not all my references. I probably have like 25 more. These are just the ones I did when I presented this to the class. But that was a lot. My brain hurts. I'm sure y'all's brain hurts. If you guys have any questions or anything like that.
You did that?
Yeah. Cool. Thank you guys.
Research.
Well done.
It's a lot.
It is a lot.
So I got a question.
Okay.
I'm just trying to think of like applicable arenas of that sort of research. Things that we can sort of control, monitor, and prescribe. Can you? One of the things that I was sort of trying to wrap my head around was that study in the idea of the volume versus intensity for that adaptation and the citrate synthase activity. Can you explain that on a slightly dumber level?
Yeah. Basically, from sprinting at super low volumes, we get just as much adaptation of that one specific molecule that you can get from an hour and a half of running. So now we've taken all that work, we put less miles on the body, and we're getting the same results. Different modalities, but citrate synthase shows us the glycolysis pathway, which is what we mainly use for exercise. So if I'm taking an exercise that's not even using that pathway in sprinting, then, and I'm still getting the same level of activity from an exercise that mainly uses it, that shows me that there's a lot of bang for buck because the system is still working the same. So if I'm explaining that to a kid, it's, hey, if I run this fast, it's harder than running for long periods of time, and my body doesn't like that, so my body will make recovery, make more effort in recovery. Does that make sense? That would answer your question, or?
Yeah, I think so.
There's a study out, well, that's old. It's kind of like the origins, like if you look at like Tabata, there's some, to that same point, is like the bang for buck, but it's like there's a certain stimulus, which obviously that's more on the biochemistry side, that allows you to hang on to more of the aerobic traits without actually doing mileage upon mileage.
Because like when we think of like aerobic exercise, we don't allow a lot of time for recovery.
Right.
Whereas sprinting, so in aerobic exercise, it's all the mindset is in time of activity, which makes sense because we don't stop, versus sprinting where an argument's like, well, you don't work out for as long, but it's like, okay, I don't work out for as long, but my body has to work a lot harder to recover from the demands I just did, the intensity. So that's what that study really was showing. Ultimately, at the end of the day, volume does win out. If I sit there and I go sprint someone 3 times versus sprinting them, 15 times with proper rest, they'll get more adaptation with more volume, but it's not as clean cut as slow pace versus fast pace. One's better for adaptation.
And we're talking like aerobic adaptation.
Yeah, aerobic, like the recovery system, not the system of replenishment in activity, but it's the recovery,
So
That's,
you get the aerobic adaptation.
yeah, so think of it like if I have a basketball player, if I have a soccer player, and they're doing constant back and forth movement and stuff, ultimately what wins out in sport is fast, quick movements. So if I can teach my body how to be as explosive, as efficiently as possible, and during my training, I've taught it how to recover as efficiently as possible, I don't have to go run 1.5 miles. I don't have to go run 2 miles because I already got all my adaptation in my 4 to 5 sprints as long as it was proper intensity.
Now, what does that say or imply, or what do you sort of intuit about the details of what that rest looks like? Is it all out rest, lay yourself down on the floor?
No, there's studies out there that show, David Bishop also had another study where it showed that if we sprint more, even without, with every sprint that we do, it gets slightly less in the phosphocreatine system and it gets more and more aerobic with each one. If we tell everyone to sprint full on capacity, that first sprint and they're warm and everything, that's going to be most intense. That's going to be the very most specific rep towards that one system. And every single time we do it, even if we give them 30 seconds, even if we give them 45 seconds, it's not the exact ratio that we're looking every single time unless you sprint once every 5 minutes. But with those short amount of times, that high intensity, I cut volume out, but I'm still getting really good adaptation. So if you get someone in here for an hour and they got to get in shape, and you have a lot of strength work you got to do too, you can get them in shape just by sprinting them 4 to 5 times, in my opinion. And then you can go work out too. So sprinting is really efficient. That's the most easiest way to say it. And when it comes to,
would it be less aerobically adaptive if you did wait that full 5 minutes between sprints? Is that sort of
no, because it's in times of epoch.
Okay, so is that because you're going through that in the rest and recovery, that's happening no matter what?
Yeah, because of the high intense of exercise, our body's in such a bad state when we stop, the initial
the initial part of all that, the other two systems kick into high gear as quickly as possible. The more we use one system, the more we have to use other systems.
The studies that you've read, what populations did they use?
That citrate synthase one, that was in trained males. Man, I kind of all over the board, to be honest. Some of that, there was some stuff with high intensity counter movement jumps and stuff that I started to look into the phosphocreatine system with like professional rugby players. That's where they were showing that those high level things. Really where I based my information on was not so much of studies where they like looked at these levels, but more of, okay, read a study, what are specific hormones that they're looking at, and then I went and read the history of all the hormones and how they kind of connect to make these pictures together and everything. A lot of the lactate stuff was done originally in rats, and then they've been starting to do it a lot more with trained men and women. And this was such a broad research thing. I guess I could, if I were to go back and I was to specifically dig into like one hormone, I could definitely dig into a specific study demographic as far as the participants, but they all showed really similar trends. Essentially, if it was untrained men and women versus trained men and women, it's just the extent of all these activations that kind of go into play.
Right. I mean, I think just like everything else, I personally still think it depends. I'm interested to go read a little bit deeper. If you have a pure sprint activity, that makes sense. You know, I think there's a place, just like for everything, a time and place for. I'm big on I'm a big, even at track practice, I'm a very low volume type person. I do think fitness qualities do improve, especially on your lesser trained athletes. But then when you start to look at your field-based sports, your court-based sports, it's like, okay, well now, there's this dynamic interplay of everything else. It's like, okay, we can get in shape, but what about tissue tolerance? What about the coordinative aspect, the actual physiology of the eccentric control and things of that nature. It's like where do we need to get the rest of the the nuggets for the actual sport from?
And I think that's why it's important as practitioners to, Lauren said it before, is like, for us to be really good at this, we have to know all our general sciences. Right. We have to know where they come into play. Right. I can be up here, and if I didn't know anything about tissue tolerance, if I didn't know anything about sport ratio, I can sit there and I can lecture you that you have a sprint, but it's like, if you ask questions like, well, what if my athlete's not even living this, you know?
And we all kind of understand that. It's like, just like on anything, in any talk, we can all go to a talk like, shit, this is it.
Yeah.
Fuck it, we're not gonna do anything else but 5 sprints, and it's like, well, yeah, how does that play into it?
I agree. I think it's just important to remember that this is just one piece of the puzzle, and then just kind of continue to learn where this fits in. If I were to take anything away from this, like I said, bang for buck, especially if I'm working with an athlete that does sprint in their sport, and they do have levels of it, because even if we just stop completely, those ratios switch. But it's also the same thing if I'm playing soccer, playing basketball, maybe I sprint and I'm just in a slow jog after that because I'm in transition or something. It's like those two systems, they're still flipping the ratios, even though we're not fully at rest. And it's just like all finding that balance, it's like, in this specific moment, how much energy am I using for here? Now, how do I recover it, And so, I guess the point I wanted to make today was, 1, bang for buck. 2, just know that everything's pretty complex, and we can't allow people to just kind of tell us it's as simple as possible, because the body works in harmony, it works in symphony with each other. All 3 of the energy systems, it all flips based off of the exact moment of demand of exercise. Cool.
You mentioned something that I'd never come across. I thought it was interesting, which is that HAF1A, the hypoxia inducible factor. would you say, appears and increases during or after sprinting because we are effectively in a hypoxic environment or metabolically?
Mm-hmm. Yeah, it increases in activity because we don't have oxygen then. The second we stop activity and we reintroduce oxygen, that's where the environment flips, and now the half-life kicks in.
That's when that clock starts to tick.
Yep.
Okay. How do you think that would be affected by differences in oxygen consumption, like increased oxygen consumption if someone's like, breathing higher percentages of oxygen, or less than that if you're exercising at altitude, or if you're even doing hypoxic work.
I think that just depends on your intensity, because workload doesn't always equal intensity. If I sprint 15 miles an hour at sea level, and I'm used to sea level, that might be my 75% intensity. So all the ratios all change. Not one system is turned off, another one's turned on, the ratios just change. Whereas if I come to altitude, now I sprint 15 miles an hour, but because of the lack of oxygen, the intensity is going to increase. So based off the intensity, that's where everything starts to kick into play too here. Living at altitude, we're already in a more anaerobic state than we can be anywhere else. And our body's already become adapted to that. But either way, 90% of intensity is still 90% of intensity. Doesn't matter my speed. If that's all my body can handle that day, then that's the adaptation that's going to occur. Those adaptations will change on other things.
And that HIF-1A, the adaptations that it causes, you said, are more like
angiogenesis, so the buildup of capillaries.
Your cardio system, your cardiovascular system itself, on the physical level.
Mm-hmm.
Okay.
Yeah, because like when we do stuff like exercise, we damage cells. HIF-1A was the ubiquitous factor of all this. So we said ubiquitous factor is like a dishwasher. Comes in, it cleans off all the damage of the cells to allow protein synthesis to reoccur. Thanks, Augie.
Now, is that only related to high intensity oxygen deprivation, or if you would say do like a long breath hold with zero intensity physically, would something like that also start to appear just because of a general hypoxic environment?
I think it would be a little bit different because cellular demand's not there. Okay. You know what I mean? Like it might be activated a little bit, but it won't be to the same extent.
Just because the oxygen consumption.
Yeah, just because we're in hypoxic versus normoxic. Yeah. Yes, those things will change, but at the same time too, you also got to remember is there's a cellular demand. If I'm just sitting here and I hold my breath, that is way different than me sprinting 18 miles an hour, with demand and not, doing that.
Yeah.
And I don't think that's a stage that's long enough. There's not enough stimulus there for me to hold my breath. By the time I actually stop having oxygen and things really start to break down, I'm going to breathe again, and all that's going to happen. Not enough HIF-1A probably is activated.
So it's more of a chemical marker, like in the blood, of how much
I think so.
CO2 ratio you
have.
have.
I think so. It's like the same thing. Like, what if I hold a muscular contraction? I just hold it for a long time under tension, but then I go and squat, and it's the same amount of time. If I squat and I have that muscle under tension for 5 seconds, and I hold an isometric for 5 seconds with no force or anything, those hormones are probably going to be a little bit different, even though the time of tension is the exact same thing.
Okay. Interesting.
That's just my hypothesis. I don't know if that's a secure answer or not, but based off of demand, usually that's how ratios all change in the body, depending on whatever hormone we're talking about.
Yeah, and I've from what I've seen from experience of non-high intense breath holding. It's more carbon dioxide buildup. That's that sort of trigger for breathing versus you actually being hypoxic.
Yeah. I now get where you're going with this, swimmers and everything like that. So that makes sense. If for me to give you a better answer on that, I'd have to dig into the different modalities of things that happen.
Yeah, that's a road I've sort of beaten down a little bit, but I've never really looked at the implications of the adaptive qualities that come from it.
One thing you probably do is look at if there's any research out there, if there's a difference between people who lift weights while holding their breath versus people who breathe when they lift weights. Getting that ventilation, getting that Valsalva's maneuver, the bracing of the core using your diaphragm to, do all that. There's research out there about breathing techniques in general. You can probably find some stuff if you really wanted to dig into that.
Cool.
Anything else? I talked y'all's ears off. Thank y'all for listening.
No.
Yeah, thank you, Dylan. That was cool. That was
Yeah.
very interesting.
All
I
right.
felt like I was back in school.
Yeah.
And I wish I was in exercise physiology last semester.
Right? No, I've gotten back to exercise. So that's-
You got your grade back?
Yeah, I got a on that I got a